Very good evening, everybody, and welcome to another episode of Eclectica, the Different News. Today, I have the founder and CTO of Dianox, Christian Code. Welcome, Christian, and thank you so much for joining me on this podcast on YouTube and LinkedIn. And I'm really grateful to you for accepting my invitation. I think we will have a lot of understanding about aptamers and related stuff in diagnostics and therapeutics in this conversation today. Thank you again.
Yeah, thank you for the invitation. I'm looking forward to talking about all the things that we could talk about aptamers and a lot of other things in science.
Right. So before we begin, Christian, can you please tell us about yourself, your journey, and how you landed up with Dianox? And that is something which is very interesting for all of us.
Yeah, absolutely. It's always fun to tell about how you ended up here, and it's quite a journey. So I'm originally from Canada, from the west coast of Canada, Vancouver. And I started in doing biochemistry just because I love the way proteins interact with different things, and how different chemical reactions enzymatic reactions can happen in the body as such. So I finished my bachelor's in biochemistry there, and molecular biology, and at the University of Simon Fraser University in Vancouver, Canada. But between that, I also sort of did a lot of work with lipids. So I worked for a small a biotech company where we made liposomes, and I developed a lot of different liposomes for three different cancer drugs at the company, and then worked in a lipid lab, and then ended up getting a Marie Curie fellowship to go to Helsinki, Finland. And it was an early stage research position, and I ended up doing my master's and PhD in biochemistry and biophysics, and working with like lipids and lipid protein interactions. And I eventually found my way to Copenhagen as a visiting scientist, and then started a postdoc position where I was again working with lipids and lipid nanoparticles. But then it was just before COVID. What happened was a guy who I met. I was in this group in Copenhagen. There's this like really cool volunteer groups where you could join, and they talk about entrepreneurship and developing your own company. One of these groups that I was in is called Rebels, which is called Rising Entrepreneurs in Biobusiness and Life Science. And there's other ones called Synapse, and a couple of different ones in the region. And Rebels really focused on helping people develop companies. And so I'll also talk a little bit about that. So we had this program with Deloitte, the big consulting firm, and we like got ideas generated and stuff. And then anyway, after that, one of the guys from Deloitte asked me, hey, do you want to start a company? And I'm like, not really. I'm finishing my postdoc. I still, do and working at this other company. I don't really want to start a company, but he was very intriguing. And at that point, he was looking at, starting to look at hepatitis B. And I was like, okay, well, you could test, hepatitis B with antibodies. But there's this thing I'd never heard of in my biochemistry courses and whatnot. And I was like, oh, these things called aptamers, which are small DNA or RNA that could fold. So they're single-stranded and they could fold into a 3D structure. And I was like, okay, this is really interesting, because then you don't need to sort of develop specific antibodies. You don't have to have something that binds the hepatitis B surface antigen. You could actually just use these specific strands of DNA or RNA that could fold, and then they could bind to this. So I was rather very, very intrigued. And then you could look at using computational models and and so it has like a lot of versatility, both in the diagnostic space, but also in the therapeutic space. And there's so yeah, so that's sort of my journey. And I've been at Dianox, so we been building Dianox for 4 years, 5 years now, and it's been quite uh, quite fun. And we've gone through different hurdles, and we developed different computational models, and developed different types of diagnostics, and now we're working on a therapeutic.
Thank you, Christian, for sharing your fascinating journey. And I mean, it's wonderful to know how you were lipid bound all the time throughout your
Yeah,
career. Yeah.
yeah.
So you told about aptamers. Is it like more in the space of precision medicine that we talk about nowadays a lot, uh,
Mhm.
or it has a generic application also?
Yeah, so I guess you like they're very similar to antibodies. So
Okay.
uh, you could have like a very specific bind to a very specific site. Bind to TNF alpha, for example, or bind to specific membrane proteins. So it's more if you were to you know yes, it is very precise, and they're very specific. And if you look at the sort of the molecular space, like we have, you know, massive database. I think the biggest database in the world of aptamers. And then you look at the molecular space, and it's in the order of trillions. So there's more. We have in our database, we could potentially have uh, more different sequences of aptamers than stars in the sky. It's not the same order of magnitude as, say, like a chemical structure where you can have sort of many different types, because there's four base pairs and they fold in this specific thing, but it's still on the order of trillions. So that being said, it's very specific for. It can be very specific for certain types of proteins or certain types of small molecules. So it's very, precision medicine or precision diagnostics. However, it's not. You potentially could sort of make something so it's you know precise for your own body. For example, if you express specific proteins, and then you have an aptamer for that. And I think one of the reasons why we started developing this is because during we started with hepatitis B, but I mentioned COVID-19, the pandemic came out. There was very few or no tests available, and we thought that it could be possible to make a test a lot faster, given if you know the protein sequence or the of some of the surface antigens of the spike protein or the nucleocapsid protein.
Okay, that's very interesting. So you say that we can use aptamers both in the diagnostic space and therapeutics. So
Yeah.
is it like, is there a position of aptamers where we can do something like targeted drug delivery for cancer in the space of oncology or any other such debilitating disease? Can you enlighten on that part if there is application there?
No, absolutely, absolutely. So different membrane proteins are on the surface of on specific types of cancer, like leukemia or whatnot. And then there's different membrane proteins, and then you could have the aptamer directly go to that membrane protein, and then you could, and back in the what we are doing in the liposome times is I also have a recent article on this where we used antibodies instead of aptamers in this case. But you could attach an aptamer very easily to a liposome, and then you could load the drug, put drugs within the liposome, and then have it go to the um, to that membrane protein that you're interested in targeting. And it's very precision medicine, where it's they like to talk about the magic bullet, where it's binds just to that site, and then goes inside, internalizes, and kills the cancer, rather than killing other things. You can also just have the aptamer loaded with drugs, so you can actually have vancomycin or sorry, not vancomycin, vincristine, for example, in the minor groove, or some other types of very insoluble, less soluble drugs in the DNA, and then that DNA or RNA can bind to the membrane protein, and then be internalized and hopefully kill the cancer. And there's a lot of different sort of modalities or different ways you could use these aptamers. And I think we haven't explored everyone. Currently there's only 2 or 3 aptamer drugs on the market, and they're mainly for the eye. And I think other people are using them, or trying to work with them more systemically.
Thank you. Thank you for answering that question and sharing those insights. I think these are really valuable and new information for many of us. Another thing which was running in my mind while you were speaking, is about biosensors. So do we have you know anything with aptamers that we can increase the biosensor accuracy or something like that? Because I think when it comes to medical device development or any kind of scientific device development, sensitivity, specificity is something which we look forward. So do you think that this space could be occupied by aptamers, or it has applications in increasing the biosensor accuracy?
Yeah, no, absolutely. So we're working on a couple projects for biosensors. So you could have electrochemical biosensors, or you could have sort of lateral flow strips. The classic, if you think of the birth, like pregnancy test, for example, and not just in accuracy, but also in like things like shelf life, or specific types of proteins or small molecules that are less available, or harder to get to with antibodies.
Right.
So I'll answer the first question. Yes, you can make them potentially more accurate by increasing the surface density of the aptamer, and then you can have more aptamers in that space compared to antibody. And the real beauty of aptamers are that you have the sequence, and then you could sort of use different computational models to fold that sequence, and then you can see where that protein or small molecule or potentially cell binds within that sequence. And then what you could do, and this is much harder with sort of antibody technology, is you could like truncate the aptamer. So you can cut it up, or you can add different regions. So that being said, you can actually make them increase the sensitivity in different ways by changing these. Additionally, you could add different groups to the 5' end or the 3' ends, or within the aptamer itself. So methylene blue is one of those groups where it acts as a redox sensor, or you could add a number of other types of things to the aptamer to increase the sensitivity. Yeah, so I think that's the real beauty of aptamers over antibodies, and that's why I think I really like it. I think I mentioned a little bit in order to get antibody, and we usually just compare them to antibodies, you have to inject a toxic substance into some kind of animal, usually like a rabbit or a horse, right? And then you get an antibody response. And then you maybe get polyclonal, or potentially try to get monoclonal antibodies from this. And then those antibodies bind to a specific protein or small molecule. But if the protein or small molecule either kills the animal or is not immunogenic, it's much harder to get that response. So in the case of back to the case of COVID-19, if COVID-19 killed the cells or killed the animal before it had time to have a specific response, it's much harder to get that antibody to be used in tests. And then the scalability is much different, a lot different because you can synthesize aptamers, and there's batch to batch, probably little batch to batch variation compared to antibodies, um, and you can use them in different places. So we're actually looking at working with aptamers in space, and some other companies have used that as well. So because it's a single, 4 base pairs, and then you could actually
Right.
print these off in space and then use them as diagnostic tools. I think there was a company called, I think they're called Aptamer right now. They actually did this in space, and there's some different trials to actually work with this in space. And for longer missions, that would be actually a really cool use case.
Okay, okay. Thank you, Christian, for sharing that information. Um, I was just trying to understand that when you tell diagnostics, like for us people who are from the healthcare domain, when we say diagnostics, so is it
Mhm.
like certain tests that we can develop with aptamers better than using antibody technology? Like for example, you said about. I mean, I'm talking more about the point of care testing area. Is there an application there, or it
Mhm.
is more into not into that space, or kind of in the helping in developing of such technology? So I just want clarity on that part, if you can help us.
Yeah, exactly. There's a very nice aptamer that binds to cocaine, and if you have people that are, if you want a really good diagnostic to look at the cocaine and cocaine metabolites, you could use this aptamer, or maybe 2 different aptamers, and then if you want to see, how if this person is using this, you can actually do that. But if you have like there's aptamers that bind to specific cancer antigens like HER2, and then you can use that aptamer to detect the specific antigen or carcinogenic CEA, carcinogenic embryonic antigen, and then you can see whether this person has a higher amount of this expressed protein. Um, Compared to yeah, there's always comparisons to antibodies, but it's really like whether you want. I think what you can compare to actually is like whether if you have some sort of mutant form. So you can actually have aptamers that might be more specific to specific than antibodies to specific mutant forms. But on a whole, they're largely comparable. But they're just a different group of binding. So I think so, and the you can put them into ELISA plates or LOSA plates in this case.
Right.
These chemical-based sensors, lateral flow strips, a number of different other techniques.
Okay, I think I got a lot of understanding from you in this aspect. So, Christian, a kind of little futuristic question, where do you see the applications or aptamer technology as such going forward in the space of
Mhm.
diagnostics and therapeutics in the say in the next 5 or probably a decade, on the next 5 or 10 years? Where can we see this technology going towards? I think understanding from you would be great because you are working in that space very, very much. Yeah.
Absolutely. The number of research articles that are being published on aptamers is increasing exponentially since it's started in the 90s. But more recently, as I mentioned, there's a lot of clinical trials that are going on. Hopefully, there's a lot more drugs that are approved in the next 5 years for aptamer-based therapies. or therapies using aptamers, like targeting cancer drugs or whatnot. And then there's a number of different biosensors that are being developed with aptamers already, and some are already on the market. And I think that in the next 5 years, there's going to be a bigger flood. I think of these biosensors, but I think there needs to be more adoption by the bigger companies, like Thermo Fisher or some of these companies that make these different plates or make these different types of things. But we're working on that to try to simplify the process of adding aptamers to these 96 well plates for Alosa plates or to different types of lateral strips. One aspect that we're working on at Dianox, and we have as I mentioned, the best database, but what we've been working on is a large language model. And I guess you're familiar with ChatGTP.
Yes, yes, absolutely. Yeah.
And there's like the Google's platform, which is called BERT. And
Yeah.
there's a number of papers that uses this platform. So we created Aptamers, which you can take any protein on Uniprot or any chemical in PubChem, and we could potentially find an aptamer for those chemicals. So, and this is done all computationally. And so you can take, a protein that's of an antigen that is very rare, and then we could go, okay, well, we might be able to find an aptamer for that, and then bind to that. And then that cuts out a lot of the work in injecting this specific antigen, which is in very small amounts, into a horse or a rabbit, so you can actually find that antigen, aptamer for that antigen. And like I said, with COVID-19 or potentially another pandemic, in the future, you can actually have aptamers that can be attached to lateral flow strips, so you can get in real time diagnostics, rather than it expanding to other parts of the world before it becomes a full on pandemic. You can actually sort of localize it and have it controlled, at least, using this.
That's fascinating. I mean, what you said, I understand that it's more of like a kind of novel AI platform where people can, come and putting it in a very, very layman language, that people can come and search for their specific aptamers, maybe particularly in the field of applications in
Mhm.
diagnostics and therapeutics. So I think that that's very interesting and that's wonderful. So uh, we are almost at the concluding part of our conversation. Before you go, I would also like to understand that, like, if it is feasible for you to share, is there any specific or exciting project that you are working on, or that might be shareable over a public podcast? So can you tell us about that, and what excites you keep going in Diagnose, and where you want to take this forward? Yeah.
Yeah, yeah, absolutely. One of the exciting things I've been working on, with a number of students, and that I could share. So I have other projects, but it's creating a number of aptamers that could diagnose different hormones or vitamin deficiencies in saliva. So
Oh,
you can
wow.
actually
Fantastic.
basically it's like spit into a tube and then see whether you have a deficiency. And a lot of there's and we did some tests early on in the lab. We found a lot of people are deficient in hormones, specific hormones, due to changes in age, or deficient in specific vitamins due to dietary differences. So having a very simple test that could to do this using aptamers, we can actually have developed this, or are developing this.
Oh, I think you struck the chord because I think whenever it's saliva, I get very excited. I am a saliva enthusiast, by the way, and I really, I think we need you one more day to talk about, about saliva and aptamers, the kind of work that you are developing. I mean, that's wonderful. Christian, thank you so much for being here on the podcast and sharing all about Dianox and the kind of work you are doing. It's groundbreaking, and I'm sure that this is going to change the near future very very soon when it comes to diagnostics and therapeutics. What would be your one message to the aspiring entrepreneurs or people who are already entrepreneurs, started out just like you with some idea, and they want to grow in this space, in the space
Mhm.
of biotechnology and healthcare? So what would be your message to them?
Yeah, I would say be curious and read. Those are things that I have done in my lifetime, where I'm like very excited about things, because I'm wondering the deeper truth, and then I end up reading a lot about these things, and then becoming an expert, and then continuing onto that journey. I was never an expert in aptamers to start with, but I'm becoming slowly better at becoming an expert. So curious and reading.
Thank you, Christian. Thank you so much once again, and namaste from India. And
Yep.
I hope we will meet very soon in another conversation. And thank you once again for your time. Thank you, everybody,
Absolutely.
for listening to this podcast. Thank you.
Yeah. Thank you. Bye.